Now it's recording.
Okay, so it is my honor to introduce today's guest speaker, Dr. Niculina Musat, a colleague and a good friend of mine. I'm going to just briefly highlight a little about Niculina's academic career. So Niculina started her studies in her homeland of Romania, completing her bachelor's in biology and her master's in microbiology and microbial genetics from the University of Bucharest. Later, she continued on for a PhD at the esteemed Max Planck Institute for Marine Microbiology. And during her time at MP, I'd say that Niculina, you have developed, in my opinion, one of the most important breakthroughs to the field of marine microbiology. And that was your HISH-SIMS approach, a method that enabled us to match phylogenies to metabolic activities with incredible precision. You have continued to be a leader in the field of single-cell methodology development, which has led you to your current position as the scientific head of the single-cell nanosims unit at the Helmholtz Center for Environmental Research in Leipzig. It is such a pleasure to have you here with us. I thank you for your time, and I'm most excited to hear your research. Welcome.
Thank you very much, Rachel. It's also a pleasure for me to be here. Thanks for the invitation. I would like to talk today to you about tools to analyze the function of microorganisms. And actually, I will going to talk about single cell tools and how one can do correlative and chemical microscopy to analyze single cells in environmental samples. So before I start with showing you the tools and explaining the techniques to you, I would like to say a few words and a few general things about the microbial world, which I'm pretty sure you already know. So you know that the vast majority of life on Earth it's microscopic, and you know that they can be found in tremendous numbers. They also make a major part of our biomass on Earth, and the diversity in human and animals, it's about 1%. So the rest of the diversity, it's out there. It's basically waiting for us to discover them and to understand them. So terrestrial, marine, surface, and subsurface habitats, and where these microorganisms are actually governing the element cycling. So as a role of microorganisms, the role of microbes is really broad. And here are just few examples to highlight how important the microorganisms are for the health of humans and the health of our planet, actually. So you probably know already that cyanobacteria and algae are very important photosynthesizers, and they are major sink for CO2 greenhouses, and they make up half of the primary production on our planet. You know probably already that microorganisms are known to degrade a lot of naturally occurring compounds. And as an example, I give you a well-known methanotrophs, which are actually able to remove the methane escaping from the seafloor. And last but not least, actually, microorganisms are able to degrade a lot of man-made compounds. And here I just choose to show you an example or give an example about the degradation of pesticides, but probably you already know that there are many projects and a lot of ongoing research on microorganisms which are able to degrade pharmaceuticals, which are able to degrade cosmetic products, which are able to degrade microplastics, which is a big issue at this point. The role of microorganisms is well known also regarding gut microbiota in nutrition and health. So these are just few examples to emphasize the importance of microorganisms and put them in a light, explaining why it's actually important to look at them, to understand their function, and why it's important to look at individual cells and not look always at the whole community. So many key questions still to be answered in microbial ecology or environmental microbiology. And when people are in these fields, they are usually interested to answer questions regarding the identity of the microorganisms or specific groups. in a certain environmental sample, how abundant they are. And of course, they are interested in the metabolic activity and function of those microorganisms in certain habitats. Structure, it's another point where many people are interested to find out if organisms are living associated with other organisms in tight associations or loose associations, attached to organic or inorganic substrates or as single cells. And also a very important point where people are like us, scientists are interested to look at is the ecophysiology. So how the microbes are influenced by the changing environment and how they can influence back. So in order to answer such questions, at least one of those, but in general, we as a scientist in microbial ecology field would like to answer at least 3 out of 4 questions. It's what type of approach we are using. So we need to consider always that the microorganisms are very small in size, in average about 1 micron. They are very complex, living in complex communities. Like I show you here an image. This is actually a picture from an oxic lake in Switzerland. And what you see here is just a drop, so a couple of tens of microliters of water from the lake. So you see a complex community of different morphotypes of microbial cells that we don't know who they are. We have no clue about their identity. We know they are in large numbers, about 10 to the 8, in this case, cells per milliliter of water. They perform different processes, biochemical processes, and sometimes these processes take place simultaneously in time. They are difficult to sample and analyze as individuals, because sometimes they form clusters, like you can see here. And what is important is that most of the microorganisms from the environment are very difficult to cultivate in laboratory, that you can find out more about their function and activity. So the tools we are working with, and I will going to present today, it's about the correlative and chemical microscopy and how one can use this these methods, actually these microscopic techniques, to find out, understand microbial cell function and activity. So what actually these tools, microscopic tools, provide you with? And here, although I know you are focused on marine environments, I just choose to give you an example from soil, which is extremely complex. I don't say that marine environment is not, but you have a really huge number of microorganisms per gram of sediment, so close to 10 to the 12. And they are most of the time living associated to inorganic and organic particles, and also with fungi, which have huge networks in this soil. So when you would apply some correlative and chemical microscopy analysis, what can you find out in such a complex environment? And what you can understand, it's spatially resolved function. So this is, and how this is happening, it's actually, first of all, you visualize and you see, like here, for example, this microbial cell in the left side, it's encrusted in iron oxides or iron sulfides. So you already know that it has a high potential to be involved in metal cycling, already iron oxidation or reduction. And it's the same here down, where you see microbial cells through a lot of precipitates of iron arsenic sulfides. So you already have an idea about what these microbes might do there. The same with the microbes here, which are associated with the fungi hyphae. So you can guess, and you can go further to investigate that these microbes have probably together a trophic interaction. By molecular mapping, molecular targeting their RNA by fish, for example, you can know which groups of microbes are active and which are not active in this environment. So you see this type of information that can point out to particular microbes involved either in nutrient cycling or maybe carbon sequestration or even degradation of various compounds. So already seeing is believing, but it also gives you hints about the potential function. And now I just give you an example what is actually correlative and chemical microscopy for you to understand which type of information you can receive when you analyze a sample. And I choose again to give you a sample from the soil. So here I start showing from the left side, it is actually a piece of soil. It is so-called rhizosphere soil. So that means it's the soil surrounding a root. It's collected as it is, undisturbed. So we try not to disturb any type of chemical gradients, because if we want to do chemical microscopy, we want to keep all the components and compartments in their place. Organic, inorganic, and microbes and roots and soil particles at the same time. So what we do, we basically embed it in resin. Yeah, we have slices, and then you have different type of microscopy from the low spatial resolution to high spatial resolution. Here you see light microscopy, yeah, overview image, then a closer look, then fluorescent microscopy. We spot a root fragment. We are interested. Particularly our scientific questions is what is happening around these root fragments and how the plant roots contribute to the transport of organic carbon in the rhizosphere. Yeah? And then the next image, it's a HIM image, it's a high resolution image to spot the same section. And further on, we go to the nanoscale resolution instruments to get molecular, to get elemental and isotopic mapping at the root and in the surroundings. Yeah, and you can see that molecular fragments, which are indicator for proteins or for phosphorus storage or for carbon, as well as elements like calcium, which is important for plant development, potassium, carbon, and also metals can be mapped together with isotopes. And you can get an isotopic ratios of those features you want to investigate. In this case, it was the plant root. Nevertheless, in order to correlate, what you will need to do is basically to register an image on top of each other. And usually you have to use one image as a host to register the other in top. And then at the end, you get information about the structure, so the morphology of your components, plus molecular, elemental, and isotopic composition. And I will show you further two instruments which we use quite often, either complementary or correlatively. In this talk, I will show you images and studies where we use them both correlatively and which type of questions we could answer with them. So NanoSIMS, on one hand, it's very good at providing isotopic ratios and measuring isotopes and elements, but we are mostly using it for providing isotopic information. And the values we obtain with this instrument is really quantitative. So what you basically get, you get isotopic ratios of single microbial cells, and this is amazing. The other instrument, the TOF-SIMS, it's more targeted on elements and molecules, but they have to be larger than 1,500 Daltons. I will go now to present each instrument separately and describe more features of those. So how is actually NanoSIMS analysis taking place? So you have a source of primary ions, which is bombarding your sample. The sample can be a soil embedded sample, like I show you, or it can be simply microbial cells, single cells distributed on a conductive substrate. It has to be conductive, because otherwise you will have an electron cloud, you will have charging, so-called charging, and you cannot extract secondary ions anymore. So you have a primary ion gun, primary ions, which is either cesium or oxygen. For the elements, light elements like carbon, nitrogen, sulfur, and phosphorus, we usually use cesium. Oxygen is more for metals. So this is shooting to your sample. And what is happening? It's happening that the sample is destroyed. And you have secondary ions as well as secondary electrons, which are being produced, released, and they are separated based on their masses. And what you have counts of secondary electrons or secondary ions per pixel or per second. And after we count this, then you will obtain, or what is actually obtained, it's the image, a reconstruction of secondary ions, for example, here, coming from the carbon 13 counts at the mass 13, or as mass 26, because because nitrogen cannot be, it does not ionize well as single ions, and it has to be in combination with carbon. And you can even differentiate between mass 26 and mass 27, so 15N and 14N if you use labeling. So what you see here, it's actually a filamentous cyanobacteria. It's actually from the Baltic Sea. You see, you have a very small field of view of analysis, about 15 by 15 micrometer. Therefore, only 3 cells can be observed, and another small microbial cell here. And what you can have here is really isotopic mapping of single cell with quantitative power. So you can look at cells, you can calculate how much each of these 3 cells are actually taking up a certain isotope, or what is their composition. So just to say a few words more, it quantifies elemental and isotopic surface composition. It is very sensitive to surface. So what this means that the sample should not have high topography, and therefore a sediment or a soil sample, if you want to keep all the to have an elemental mapping also surrounding your cells, it has to be flat. And many times people are using resin embedding to do that. It has a small beam of primary ions, and this gives you the lateral resolution. You can detect parallel masses, up to 7 masses in parallel, and we mostly use it in combination with stable isotope probing. So this is why I show in previous image, I show you 13C and 15N, because we quite often use stable isotope labeling experiments to particularly target microbes which are able to take up a certain substrate, either carbon or nitrogen, or you can look also at the general activity with, for example, the deuterated water. So you can basically image metabolically active microbial cells, or those responsible for taking up a certain substrate. You can follow the assimilation of the label in time through the community. Of course, you can calculate assimilation rates, and you can determine in this way the contribution of a certain group to a certain biochemical processes. So
excuse me.
Yes.
So if I understand this correctly, the nanosims analysis, it basically, this PI gun shoots the sample, and then kind of like destroy it, and then these magnets will then sample the different compounds to give a picture.
Yes.
Okay, good.
The different secondary ions.
Okay, secondary ions. Thanks.
So the other tool, what we are using for chemical imaging, it's TOF-SIMS. So TOF-SIMS, it's known very well for molecular analysis. So it gives you molecular information. So what is actually this instrument doing? It's also destructive, but it's less destructive than the nanosims. And recommended if you use it in tandem on the same sample, basically, and you want to do correlative work, it has to be before the nanosims. It is more flexible because you have a variety of sources. Not only of single ions, but also of cluster ions like bismuth. So you can switch even between the sources to bombard your sample. And here is just an example of, for example, Beggiatoa alba, where we map the filaments which were deposited on a polycarbonate filter. And this is what you see. You can go in depth. You can go through the biomass, in this case, the filaments, and map different molecular fragments. And the beauty of this instrument is that you can record all molecular fragment spectrum. So you do not have specific masses like you have with nanosims. With nanosims, you have to specifically choose the mass before, and you can have up to 7 masses. Here you get all kinds of molecular fragments, like here we got combination of phosphorus with oxygen, combination of carbon with nitrogen and oxygen, even carbon alone. So you can get all kinds of combination, and you can get also a pretty good lateral resolution, a bit higher. I mean, not as good as with nanosims, but the mass resolution here, it's pretty poor. And usually it is on the expense of the lateral resolution. So the better imaging you want to get, then you have to decrease your mass resolution, and therefore the identification of your molecular fragments, it is a bit more difficult. And I will show you in the next slide what you basically get from a TOF-SIMS analysis. You get molecular fragments. And here it's an example where you have a hyphae of a fungi with microbial cells, small ones in blue. Yeah? And what type of information you actually get. Here we use a bismuth to shoot at the sample, and you see you get all kinds of molecular fragments. Yeah? And the biggest challenge here is to identify precisely these fragments as belonging to, as usually for each peak you have more, more identity, molecular identity to choose from. So at the 150 nanometer spatial resolution that you can see very well and visualize, you have a pretty poor mass resolution. But nevertheless, the instrument is pretty flexible, and I will show you that using combination with other tools can give you very important information. So now I would like just to show you a few examples how we use these instruments in tandem, so correlatively, and what we find out. And the first study case, it was about the metabolic interactions between marine organism, Acantharia, and the photosynthetic algae, Phaeocystis. So our purpose here was to look at the native elemental, isotopic, and molecular mapping, and to understand how this algae, this symbiotic algae, behaves within the host and as free living. And here I have you an example where we combine transmission electron microscopy. So first of all, the whole marine organism was embedded in resin. It was sliced in small slices, like you see here in the left corner. And with a green arrow are pointed out the symbionts within the host tissue. This is TM, also A up and down, they are both TM with a different resolution. Here you have an overview, and here you have simply just one symbiotic cell. And we perform on a B, a nano or a TOF SIMS imaging of the very same area, looking at particularly at nitrogen mapping, then followed by a nano SIMS analysis, which is C and E. And down, it's the same picture. So as I mentioned, with the nanosims, you can get isotopic ratio, and then you can basically map this within one cell. So if we go further here, the combination of nanosims and TOF SIMS looking at the nitrogen, what we could observe that particularly the symbiotic algae, when they are in the host, they are enriched, extremely enriched in nitrogen. And for us, so when you have accumulation of nitrogen in a cell, that means a lot of synthesis of proteins and nucleic acids. So this pointed out for us that this algae are extremely metabolically, extremely active. And when we compare them with the free living, I'm not showing this now, but when we compare them with free living, there was a huge difference between the activity and symbiotic mapping as element and as isotopes in the symbiotic symbionts versus the, or symbiotic algae versus the algae as free living. Another point, what we wanted to look, and the TOF SIMS is known for that, it can map lipids. So in this case, we look at phospholipids, and actually the molecular fragments you see here down in different colors, these are indicator for different type of molecules within the cell. So we could map the phospholipids, we could map the nucleic acids, we can map the proteins, as I mentioned, in nitrogen-rich chloroplast, and we have seen the localization of fatty acids in vacuoles within the host cells and in the symbiont cytosol. So based on this study, we could see differences between symbiotic and free living, and what we could conclude was that the host seems to reconfigure the structural organization, activity, and trophic microhabitat of its symbiont. Another example here, and what I want to go further, is to tell you that the best way, or the most common way that people are using the nanosims, it's in a combination with stable isotope labeling to track microbial activity in environmental samples, and in combination with fluorescence in situ hybridization. And how one would do that is to simply collect an environmental sample to perform your stable isotope probing, so stable isotope labeling incubations. The beauty of with this technique is that the instrument is so sensitive that you can basically do very short time incubations, and you do not go into problems regarding bottle effects or other things related with keeping an environmental sample in a closed system for a long time. After that, if you are interested in a particular group of microorganisms, you can use a fish probe that is binding to the ribosomal RNA and is specific for a certain group to hybridize these cells, so to see them highlighted in a certain color depending on the fluorochrome you use. You can use this sample to map it and mark it, and then go back into the nanosims and find exactly the same region and analyze those single cells with respect to their elemental and isotopic composition. And at the end, you can get the function of a particular group of microbes. In this case, it was uptake of 13C labeled glucose. It can be any type of substrate. It is important this substrate stays in the cell. If it is respired and it's outside the cell, then you cannot say anything about this process. So what you can do with this type of technique, you can look at the photosynthesis in plankton, or let's say fixation of carbon. You can look at syntrophic interactions and channeling through these partners of carbon and nitrogen. You can look at different processes like nitrogen fixation. You can look at phototroph heterotroph interactions or microbe host interaction. And as I mentioned, you can apply fish whenever you want to have the phylogenetic identity of the partners prior to measuring isotopic and elemental composition by nanosims. And here is just a short example for those of you which are not maybe familiar with the technique. When you take an environmental sample, and here I'm back at the anoxic water sample, you see a huge diversity, and the diversity is not only in shape and morphology, but it is also in terms of different phylotypes and different functions. And in order to focus on a certain group, then you would apply a fish probe, which is specific for a certain group. In this case, it was Lamprocystis, these clustering cells of anoxygenic phototrophs, and you can particularly distinguish them, mark them, and then you can go then further with the nanosims analysis, particularly focus on this group of microbes if you are interested in their activity with respect to uptake of a certain substrate. The other cells, you can simply use a DAPI staining just to see them, but the phylogenetic information is given by the fish hybridization. Nevertheless, when you have a simple one-to-one system, or when you can identify your partners based on their morphology, even if it's not one-to-one system like this synthetic system here between a fungi and Bacillus subtilis, you do not need fish. So even if you have more than one partner, but you can clearly identify them based on morphology, therefore fish is not has to be used before the nanosims analysis, because you know the identity of the partners involved. And here I choose to give you an example of a trophic interaction between a fungi and bacteria. It was a synthetic system created in the laboratory, just to give you another example how one can tackle several different questions with the chemical microscopy. So what we basically did in this study, we let the fungi to develop from one site to another, crossing a small silica wafer where we have spores of Bacillus subtilis. Our specific question was, can fungi hyphae carry, for example, water and nutrients to the spores? And if the spores in the surroundings were the spores where the hyphae are crossing, can actually make use of these nutrients and water, and then they can develop maybe in vegetative cells. So this is the, in the left side, you can see how the fungi developed over the silicon wafer where we had the spores of Bacillus subtilis. They created a lot of hyphae. And with a closer look, with a light microscopy, we have seen that a lot of the spores were actually transforming themselves in vegetative cells and were aligned very nicely parallel with the hyphae. Nevertheless, this experiment was done in combination with the labeling experiments to see how carbon, nitrogen, and water is carried from the hyphae to the Bacillus subtilis. And the first analysis we've done in this case, it was a TOF-SIMS analysis. And you can see here various images of different masses. So here you have what is in this overlay image. You have the phosphorus, you have the sulfur, and then you have the CN. And what you can see is that you see a lot of green developed vegetative cells, and in blue you always see the spores which you also see in the F and this is the sulfur. So the spores had a particular signature in sulfur, being very enriched in sulfur, and therefore very easily spotted. Here we perform the TOF-SIMS analysis just to get an impression where the hyphae are positioned, and we could see that by the CN signal, and where the spores and the vegetative cells are located, to basically spot areas that will be further interesting to analyze with nanosims. Because nanosims, I explain you, it goes in very small fields of view, while TOF-SIMS can scan larger areas on a sample. Nanosims in this case was used on specifically to look at the uptake and transfer basically of the carbon, which was given in a form of 13C glucose, ammonia, and 18O water to the spores, and basically to the vegetative cells. And what we could see that, of course, is no big surprise that vegetative cells would be enriched, and clearly they got from the hyphae, because in the beginning of the experiment only hyphae were labeled. So the transfer was clear, and this is not a surprise. What it was actually surprise for us, or let's say what we discovered with this study, it was the special context. So what we could see is that the spores which are further away from the hyphae, starting on 1.7 to 2 micrometers, they are not enriched anymore. So the leaking of the nutrients and water from the hyphae, it went up to maximum 1.72 micrometer in the surroundings. So those spores that were by accident being in the way of the hyphae, and close to this hyphae, they could receive the water, carbon, and nitrogen, and therefore transform the cells in the vegetative cells, while the spores further away from these hyphae remain spores, because they had no access to nutrients and water. So, spatially and chemically resolved evidence we provide by this study that the proximity it plays to hyphae, it plays a significant role in germination of the spores. And therefore it highlights the importance of the mycelium-mediated transfer of nutrients and water also in systems like soil, for example, where they are in high numbers. So I hope that I convince you that these techniques are very important in the field of microbial ecology and environmental microbiology. I would like to acknowledge research team as well as collaborators that I collaborated in the past and still collaborating with them. My team in Leipzig, and I would like to thank you for your attention, and I'm open for questions. Even if you have questions, not immediately, but later on, you can mail them to me. Thank you for your attention.